Bioenergetics of exercise and training. Metabolic specificity of exercise and training is based on an understanding of the production and use of energy in biological systems. Efficient and productive training programs can be designed through an understanding of how energy is produced for specific sports of exercise, and how energy production can be modified by specific training programs. After defining essential bioenergetics terminology, including the role of adenosine triphosphate, ATP, this chapter discusses the three basic energy systems that work to replenish ATP in human skeletal muscle. Then we look at substrate depletion and repletion, especially as related to fatigue and recovery, bioenergetic factors that limit performance, and aerobic and anaerobic contributions to oxygen uptake. Finally, the metabolic specificity of training is discussed. Essential terminology. Energy can be defined as the ability or capacity to perform work. Various forms of energy exist: mechanical, chemical, electromagnetic, heat, and nuclear energy, for example. The transformation of energy from one form to another is essential in almost every activity. In a biological system, the conversion of chemical energy to mechanical energy is necessary for many functions, including movement. Bioenergetics, or the flow of energy in a biological system, is concerned primarily with the conversion of food, large carbohydrate, protein, and fat molecules which contain chemical energy into biological, usable forms of energy. The breakdown of chemical bonds in these molecules release the energy necessary to perform work as muscular activity. The breakdown of large molecules into smaller molecules associated with the release of energy, termed catabolism. The synthesis of large molecules from smaller molecules can be accomplished using energy related to from catabolic reactions. This building up process is termed anabolism. The breakdown of proteins into amino acids is an example of catabolism, while the formation of proteins from amino acids is the anabolic process. Exergenic reactions are energy-releasing reactions that are generally catabolic. Endogenic reactions require energy and include anabolic processes and the contraction of muscle. Metabolism is the total of all the catabolic, exergonic, and anabolic, endogenic reactions in a biological system. Energy derived from catabolic exergonic reactions is used to drive anabolic and endogenic reactions through the intermediate molecule adenosine triphosphate, ATP. ATP allows the transfer of energy from the exergonic to the endogenic reactions. Without an adequate supply of ATP, muscular activity and muscular growth would not be possible. Thus, it is apparent that strength and conditioning professionals should have a basic understanding of how exercise affects ATP, used and resynthesized when designing training programs. ATP is composed of adenine and nitrogen-containing base ribose, a 5-carbon sugar, collectively called adenosine, and 3 phosphate groups. The removal by hydrolysis of one phosphate group yields adenosine diphosphate, ADP, and hydrolysis of a second phosphate group yields adenosine monophosphate, AMP. In essence, ATP provides the energy for muscular contraction and thus human movement. ATP is classified as a high-energy molecule because it stores large amounts of energy in the chemical bonds of the 2 terminal phosphate groups. The breaking of these chemical bonds release energy to drive various reactions in the body. Because muscular cells store ATP only in limited amounts, and activity requires a constant supply of ATP to provide the energy needed for contraction, ATP producing processes must occur in the cell. Biological energy systems. 3 energy systems exist in mammalian muscle cells to replenish ATP. Phosphagen system, an anaerobic process, i.e. one that occurs in the absence of molecular oxygen. Glycolysis, of which there are 2 types, fast glycolysis and slow glycolysis, oxidative and anaerobic process. One requires molecular oxygen. Of the 3 main food components, carbohydrates, fats, and proteins, only carbohydrates can be metabolized for energy without the direct involvement of oxygen. Therefore, the importance of carbohydrates in anaerobic metabolism cannot be overestimated. All 3 energy systems are active at a given time. However, the extent to which each is used depends primarily on the intensity of activity and secondarily on its duration. Phosphagen system. The phosphagen system provides ATP primarily for short-term, high-intensity activities, resistance training and sprinting, and is active at the start of an exercise regardless of intensity. This energy system relies on the chemical reactions of ATP and creatine phosphate, two phosphagens, as well as the enzymes myosin ATPase and creatine kinase. Myosin ATPase catalyzes the hydrolysis of ATP to form ADP, an inorganic phosphate. The release of energy, creatine kinase, catalyzes the synthesis of ATP from creatine phosphate and ADP. Creatine phosphate supplies a phosphagen group that combines with ADP to form ATP. These reactions provide energy at a high rate. However, because ATP and creatine phosphate are stored in the muscle in small amounts, the phosphagen system cannot supply energy for continuous long-duration activities. Approximately 5 millimoles of ATP and 16 millimoles of creatine phosphate are stored in each kilogram of muscle. Generally, type 2 fast-twitch muscle fibers contain greater concentrations of phosphagen than type 1 fibers. Although an important reaction of the phosphagen system is myokinase reaction. This reaction provides an immediate source of ATP. It is also important because AMP is a powerful stimulant of glycolysis. Control of the phosphagen system. Creatine kinase activity primarily regulates the breakdown of creatine phosphate. An increase in the sarcoplasmic concentration of ADP promotes creatine kinase activity. An increase in ATP concentration inhibits it. At the beginning of exercise, ATP is hydrolyzed to ADP, releasing energy for muscular contraction. This increase in ADP concentration activates creatine kinase to catalyze formation of ATP from the breakdown of creatine phosphate. Creatine kinase activity remains elevated and exercise is continued at a high rate of intensity. If exercise is discontinued or continues at an intensity low enough to allow glycolysis or the oxidative system to supply an adequate amount of ATP for the muscular cells' energy demands, the sarcoplasmic concentration of ATP will likely increase. This increase in ATP then results in decrease in creatine kinase activity. Glycolysis. Glycolysis is the breakdown of carbohydrates, either glycogen stored in the muscle or glucose delivered from the blood, to produce ATP. That supplements the supply from the phosphagen system for high-intensity muscular activity. The process of glycolysis involves multiple enzymatically catalyzed reactions. The enzymes for glycolysis are located in the cytoplasm of the cells, the sarcoplasm in muscle cells. As seen in figure 5.2, the process of glycolysis may go on in one or of two ways, termed fast glycolysis and slow glycolysis. During fast glycolysis, pyruvate is converted into lactic acid, producing ATP at a fast rate as compared with slow glycolysis, which pyruvate is transported to the mitochondria for use in the oxidative system. Fast glycolysis is commonly be called anaerobic glycolysis and slow glycolysis aerobic glycolysis as a result of the ultimate fate of the pyruvate. However, because glycolysis itself does not depend on oxygen, these terms are not practical for describing the process. The fate of the end products is controlled by the energy demands within the cell. If energy must be supplied at a high rate, such as during resistance training, fast glycolysis is used primarily. If the energy demand is not as high and oxygen is present in sufficient quantities in the cell, slow glycolysis is activated. Fast glycolysis. Fast glycolysis occurs during periods of reduced oxygen availability in the muscle cells and results in the formation of the organic end product, lactic acid. Muscular fatigue experienced during this exercise is often associated with high tissue concentrations of lactic acid. Lactic acid accumulates in tissues as a result of an imbalance between production and utilization. As lactic acid accumulates, there is a corresponding increase in hydrogen ion concentration, which is believed to inhibit glycolytic reactions and directly interfere with muscular excitation-contraction coupling, possibly by inhibiting calcium binding to troponin, or by the interference of cross-bridge formation. Also, the decrease in pH inhibits enzymatic activity in the cell's energy system. The cumulative effect of this decrease in availability of energy and muscular contraction forces during exercise. Lactic acid is converted to its salt, lactate, by buffering systems in the muscle and blood. Unlike lactic acid in the muscle, lactate is not believed to be fatigue-producing substance. Instead, lactate is often used as an energy substrate, especially in type 1 and cardiac muscle fibers. It is also used in gluconeogenesis, the formation of glucose from the lactate and non-carbohydrate sources during extended exercise and recovery. Normally, there is a low concentration of lactate in the blood and muscle. The reported normal range for lactate concentrations in blood is 0.5 to 2.2 millimoles at rest and 0.5 to 2.2 millimoles for each kilogram of wet muscle that has not been dissected. Lactic acid production increases with exercise intensity and it appears to depend on muscle fiber type. Researchers have reported that the maximal rate of lactic acid production for type 2 muscle fibers is 0.5 millimoles gram, -1 second, -1. And for type 1 muscle fibers, 0.25 millimoles, same. The higher rate of lactic acid production by type 2 muscle fibers may reflect a higher concentration or activity of glycolytic enzymes than that of type 1 muscle fibers. Although the highest possible concentration of lactate accumulation is not known, complete fatigue may occur at blood concentrations between 20 and 25 millimoles. One study, however, reported blood lactate concentrations of greater than 30 millimoles following multiple bouts of dynamic exercise. Along with exercise intensity and muscle fiber type, exercise duration, state of training, and initial glycogen levels can also influence lactate accumulation. Blood lactate concentrations reflect lactic acid production and clearance. The clearance of lactate from the blood reflects a return to homeostasis, and thus a person's ability to recover. Lactate can be cleared by oxidation within the muscle fibers in which it was produced, or it can be transported in the blood to other muscle fibers to be oxidized. Lactate can also be transported in blood to the liver, where it is converted to glucose, the process referred to as the Cori cycle, as depicted in 5.3. Galnick et al. Have reported that blood lactate concentrations normally return to pre-exercise values within an hour after activity. Light activity during the post-exercise period has been shown to increase lactate clearance rates. And aerobically trained and anaerobically trained athletes have faster lactate clearance rates than untrained people. Peak blood lactate concentrations occur approximately 5 minutes after the cessation of exercise, a delay frequently attributed to the time required to buffer and transport lactic acid from the tissue to the blood. Blood lactate accumulation is greater following high-intensity intermittent exercise, resistance training, and sprints, than following lower intensity continuous exercise. Jacobs reported highest blood lactate concentrations after maximal bouts of anaerobic exercise. Stone et al. Observed that multiple sets of resistance exercise squats with increases in resistance to failure resulted in higher blood lactate concentrations in trained individuals than in untrained individuals, and that the time to failure and total work accomplished were greater in trained people than untrained people. However, trained people experience lower blood lactate concentrations than untrained people when exercising at an absolute workload, same resistance. This indicates that resistance training results in alterations of lactate response similar to those from aerobic training. These alterations include a lower blood lactate concentration at a given workload in trained individuals and higher blood lactate concentrations in trained individuals during maximal exercise. The net reaction for fast glycolysis may be summarized as follows. Glucose plus 2-phosphate plus 2-ADP converts to lactate plus 2-ATP plus H2O. Slow glycolysis. If oxygen is present in sufficient quantities in the mitochondria, specialized cellular organelles where the reactions of aerobic metabolism occur, the end product of glycolysis is pyruvate. It is not converted to lactic acid, but is transported to the mitochondria. Also transported, there are two molecules of reduced nicotinamide adenine dinucleotide, NADH, produced during glycolytic reactions. Reduced refers to the added hydrogen. When pyruvate enters the mitochondria, it is converted to acetyl-CoA, CoA stands for coenzyme A, by the pyruvate dehydrogenase complex. Acetyl-CoA can then enter the Krebs cycle, discussed in page 80. For further ATP production, the NADH molecule enter the electron transport system, discussed on page 80 also, where they can also be used to produce ATP. The net reaction for slow glycolysis may be summarized as follows. Glucose plus 2-phosphate plus 2-ADP plus 2-NAD plus converts to 2-pyruvate plus 2-ATP plus 2-NADH plus 2H2O. Energy yields of glycolysis. Glycolysis produces a net of 2 molecules of ATP from 1 molecule of glucose. However, if glycogen, the stored form of glucose, is used, there is a net production of 3 ATP because the reaction of phosphorylating, adding a phosphate, to glucose, which requires 1 ATP, is bypassed. Control of glycolysis. Glycolysis is stimulated during intense muscular activity by ADP, inorganic phosphate, ammonia, and slightly decreased in pH, and strongly stimulated by AMP. It is inhibited by the markedly lower pH that may be observed during periods of inadequate oxygen supply and increased levels of ATP, creatine, phosphate, citrate, and free fatty acids that are usually present at rest. Phosphorylation of glucose by hexokinase primarily controls glycolysis. The rates of glycogen breakdown to glucose, which is catalyzed by phosphorylase, must also be considered in regulation of glycolysis. Another important consideration in the regulation of any series of reactions is the rate-limiting step. That is, the slowest reaction in the series. The rate-limiting step in glycolysis is the conversion of fructose 6-phosphate to fructose 1,6 biphosphate, a reaction catalyzed by the enzyme phosphofructokinase, PFK. Thus, the activity of PFK is of particular importance in regulating the rate of glycolysis. Activation of phosphagen energy system and the production of AMP through the myokinase reaction stimulates glycolysis by stimulating PFK to contribute to the energy production of high-intensity exercise. Ammonia produced during high-intensity exercise as a result of AMP, or amino acid deamination, removing of the amino group from the amino acid molecule, can stimulate PFK. This effect may partially offset the results of the lower pH experienced during high-intensity exercise, which inhibits both phosphorylase and PFK activity. Lactate threshold and onset of blood lactate. Recent evidence suggests that there are specific breakpoints for the lactate accumulation curve. As exercise intensity increases, the exercise intensity or relative intensity of which blood lactate begins to abrupt increase above the baseline concentration has been termed lactate threshold, LT. The LT represents an increased reliance in anaerobic mechanisms. The LT typically begins at 50 to 60% of maximal oxygen uptake in untrained subjects and 70 to 80% in trained subjects. A second increase in the rate of lactate accumulation has been noted at higher relative intensities of exercise. This second point reflection has been termed the onset of blood lactate accumulation, or OBLA, and generally occurs when the concentration of blood lactate is near 4 millimoles per liter. This breaks the lactate accumulation curve may correspond to the points in which intermediate and large motor units are recruited during increased exercise intensity. The muscle cells associated with large motor units are typically type 2 fibers, which are particularly suited for anaerobic metabolism and lactic acid production. Some studies suggest that training at intensities near or above the LT or OBLA pushes the LT and OBLA to the right. An example, lactate accumulation occurs later at a higher exercise intensity. This shift probably occurs as a result of the changes in hormone release, particularly reduced catecholamine release at high exercise intensities and increased mitochondrial content that allows for greater production of ATP through aerobic mechanisms. Thus, shift allows the athlete to perform at higher percentages of maximal oxygen uptake without as much lactate accumulation in the blood.